Okay, guys, so anatomy and physiology is the study of the structure and the function, and both are really inseparable. You cannot you cannot separate anatomy from physiology, and you cannot separate physiology from anatomy because the structure gives us the function. So the knife is able to cut because the knife structure is sharp, and the hammer is able to hammer down a nail because it's dull. So if it wasn't for the structure of the knife being sharp, it will not be able to do the function of cutting. A police officer, walks around with a taser and walks around with weapons, but a chef in the kitchen does not have any weapons, has the cooking utensils, for example, because the job, the function of the police officer is to be dealing with a lot of criminal activity while the chef is cooking, so they need the proper structure to enable them to do the function. If we look at this in the organ system, we see that a cardiac myocyte, the heart, the cell of the heart, if you look at the cell of the heart under the microscope, it does not look anything like the cell of the nervous system, the neuron. So these are two cells, the neuron and the cardiac myocyte. So the neuron is the parenchymal. Parenchyma is the actual tissue cell. So the neuron, the neuron is the cell for the nervous system. There's the parenchymal cell of the nervous system. And the cardiac myocyte or the cardiomyocyte is the parenchymal cell of the heart. As the parenchymal cell of the heart. Both of these cells under the microscope, histologically. So histologically, these two cells, a neuron and a cardiac myocyte, look nothing like each other. So why is that the case, even though they have the same DNA? If you look inside the DNA of a neuron and you look inside the nucleus of a cardiomyocyte, you will see that they have the same exact identical DNA. So why are they looking different then? If they have the same DNA, if you look inside the nucleus looks like that. If you look inside the nucleus and you have the chromosomes and they have long threads of DNA that is double helix with complementary base pairs. This is the DNA. You look the DNA of the neuron and the DNA of the cardiomyocyte. You see that it looks identical, mirror image, the same exact DNA. Well, then why is it the case that the neuron under the microscope looks different than the cardiomyocyte? Why is that? These are two different cells. I can add to the list. I can add an islet of Langerhans. This is the cell of the pancreas. The islet of Langerhans. I can add to the list a musculoskeletal cell. I can add to the list a hepatocyte, the cell of the liver. Hepatocyte, the cell of the liver. I can add to the list a nephron, nephron, that is the cell of the kidneys. Well, these cells look different. They look nothing like each other, but they all have the same exact DNA. How did that come about? Who can speculate and give us an educated guess onto why is it the case that a cardiac myocyte is different from a neuron, which is different from an islet of Langerhans at the pancreas, which is different from the hepatocyte of the liver, which is different from the nephron, which is different from the dermatocyte, cell of the integument, the skin. Why do they all look different and they all inside their nucleus have the same DNA? Yeah, they are different structurally and they have different functions. If you pluck off, remove a nucleus from the islet of Langerhans and put it in a cardiac myocyte, it will work. If you remove a nucleus from the neuron, remove a nucleus from the hepatocyte, switch them around, it will work perfect. They will undergo cell division, no problem. They did that experiment. They brought a musculoskeletal cell from a frog, removed the nucleus. Then they brought an ovary cell, the egg, the ova. They removed the nucleus. That's the ova, an egg that has a nucleus like that. And let's say that, for example, in green, that's the frog skeletal. Obviously, this is a frog ova. If you put a nucleus of some other species, obviously it's not going to work because that's a different DNA. But we're talking about the same DNA. That's a frog ova, and this is a frog skeletal muscle. So what they did was they removed the skeletal muscle nucleus and the ova nucleus, and they switched them around. They put the nucleus of the skeletal muscle of the ova in the skeletal muscle, and they put the nucleus of the skeletal muscle in the ova. Then they fertilized. They fertilized the frog with a sperm, obviously frog sperm. And they said that it's not going to work. Their hypothesis was it's not going to work because this is not the same nucleus. This is not a frog. This is not a nucleus. I fertilized the skeletal muscle. They fertilized the ova with a frog sperm. So their idea was this ova is not going to undergo cleavage. It's not going to undergo embryogenesis. We're not going to have an embryo because, you see, this is not the nucleus of an ova frog. This is the nucleus of a skeletal muscle of the frog. Well, guess what? It divided fine, and then it developed into an actual frog embryo. So that is how they discovered that the DNA is the same in all these different types of cells. The DNA is identical in all the cells. But why are the cells different if their DNA is the same? Obviously, they have different function. They have different function, but
is it because of meiosis or mitosis? This is a class.
So they all have different function based on a different structure that these cells have histologically. But even though you see how are we talking about different and different? They have different structure, they have different function, same DNA. Different structure, okay. So these cells, guys, they have different structure, they have different function, but they have the same what? They have the same DNA. So how did we move from having the same, how did we move from having the same DNA to having different structures and functions?
Cell division.
Yeah, so the cell division happens in all the cells. The answer is just because you have the same DNA, it does not mean all of it is transcribed. So the DNA looks like that. This is all the DNA in the body. So this particular region, if you transcribe it, you will get heart muscle proteins. Heart muscle proteins. You transcribe this gene right here. That's gene number 1. That's gene number 1. That's gene number 2. That's gene number 3, for example. Number 3. That's gene number 4. That is gene number 5. And that is gene number 6. So the law says every gene encodes a special, unique protein. So if you transcribe gene number 1, you're going to get a heart muscle protein. If you transcribe gene number 2, you're going to get a hepatocyte protein. If you transcribe gene number 3, you're going to get an isolate of Langerhans pancreatic cell protein. If you transcribe gene number 4, you're going to get neuron protein. If you transcribe gene number 5, you're going to get nephron protein. If you transcribe gene number 6, you're going to get bone, osteocyte proteins. So for example, all these cells are different because for the neuron, this is the gene that's active, that is on, and everybody else is off. Everybody else is off. Only in the neuron, only the region that will give you proteins that will help the neuron work is on. Everything else is off. If you are looking under the microscope, looking at a heart muscle cell, a cardiac myocyte, you will see that this gene is on and everybody else is off. So the region that is on is called euchromatin. Euchromatin. While the region that is off is called heterochromatin. So euchromatin is on in the cell that needs it, and heterochromatin is off for the cell that doesn't need it. So what does that mean? That means if you are euchromatin for one cell, that means for the other cell, you're going to be heterochromatin. Because one cell cannot have all of the genes on. So for the heart muscle, gene number 1 is going to be on, euchromatin. Everything else is going to be off, heterochromatin. For cell number 2, the liver cell, the hepatocyte, gene number 2 is going to be on, euchromatin, and everything else is going to be off, heterochromatin. For gene number 3, the isolate of Langerhans, that gene is going to be on, euchromatin, everything else is going to be heterochromatin. For gene number 4, the neuron, that region is going to be on, euchromatin, everything else is going to be off, heterochromatin. This concept is really important. This concept is really important for you to understand that all the cells, obviously in the same organism, we cannot say that all the cells of different species. That doesn't make sense. You have to be within the same species, human beings, frogs, horses, worms. As long as you are within the same body, you have the same DNA. But the same DNA is lazy, is off. Most of it is heterochromatin. It is the euchromatin that makes the cell different. So if you get a sample of every single cell in your body, a cell from your brain, a cell from your heart, cell from your blood vessels, a cell from your liver, a cell from your kidney, You look inside their nucleus, you will see they all have the same exact DNA, but it is on and off in different fashions. The gene that is euchromatin on for one cell will not be the same one on for the other cell. My identity is my euchromatin is what makes me who I am. Each cell, it is the euchromatin that defines it, that makes it the cell. That's what it is. Yeah, so melatonin is one of the proteins of the dermatocyte, the cell of your skin. So the gene that needs to be turned on so I can get the melanin protein is only going to be turned on in your melanocyte, in the cell of the skin. It's there in the liver, but it's off. It's not turned on. It's there in the melanocyte gene, is there in your brain cell, but it's not on, it's off. It's heterochromatin. It will only be euchromatin inside the melanocyte. So when I look inside my neuron, this is really, really important. When I look inside my neuron, when I look inside my cardiac myocyte, I'm gonna find in my cardiac myocyte a lot of cytoskeletal mechanical proteins, actin, myosin, dynein. These, why? Because the job is for the heart to pump mechanical force, pressure, 120 millimeters of mercury. That's gonna need a lot of proteins, scaffolding proteins to pump a lot of blood. But will I need the same amount of cytoskeletal mechanical proteins in the neuron? Will I see that in the neuron?
No.
Of course not, because the neuron is not pumping anything.
No.
Will I see my police officer having a gun and a taser? Obviously. Will I see the chef having a gun and a taser? Same thing in the heart. I'm going to see my heart cell full of actin, myosin, cytoskeletal pumping proteins. But my neuron is not going to have these cytoskeletal pumping proteins because the job of the police officer is to deal with crime. They have weapon, while the job of the chef is to cook. So the job of the heart is to pump blood, 120 millimeters of mercury. But the neuron is not pumping, so it doesn't have. So do you now appreciate how our body is working in a balance? What is your job? You get what you need only. The euchromatin is only for what you need to do your function. Everything else off. How do we manage that? It's called genetic expression. We express the genes that help me be who I am and do my job. But I turn off everything else. And you know, this language on and off is for us to understand. But in anatomy and physiology, we have our own language. Heterochromatin, when I say on and off, that's for somebody that got nothing to do with nursing or with anatomy and physiology. But you, when I say heterochromatin, pop, you immediately know heterochromatin is the part of the DNA in the cell that doesn't need it. It's not going to be expressed. It's the one that is turned off. And when I say euchromatin, you say, oh, I know what that is. That is the part that gets expressed because my cell needs it. Is key concept number one done? Are we clear?
Yeah.
Okay. I need somebody to define for me what is heterochromatin. Yeah. So heterochromatin is a part of our DNA that is off, inactive, because
it cannot be on. It cannot be on, because it has a balance, right?
it's not needed. You don't need it. Good. Good. Very good. What is euchromatin? So the gene part, the part of the DNA that is on. So now please explain for me, why is it the case that a hepatocyte, the cell of the liver, isolate of Langerhans, the cell of the pancreas, a neuron, the cell of the nervous system, a cardiac myocyte, the cell of the heart, all of them under the microscope histologically look different. But when I look inside the nucleus, they have the same exact DNA. Switching the nucleus, taking this nucleus from this one, plucking it here, taking this one, putting it there, they will carry the function fine. Why is that the case?
Because some aren't turned on because it's not function. It's only the one that function is on.
Okay,
So the rest is like extra. Pro.
excellent, very good. You guys today, expert anatomy physiologists. Okay, all of that was concept number one. Now, concept number two. For concept number two, we're gonna learn in anatomy and physiology. So we already established that in anatomy and physiology, we're gonna be learning about genes that get turned on and genes that get turned off. And when the genes get turned on, what do they become? And when the genes get turned off, what is the final and what is the ultimate fate of the genes that are off? The next concept you need to know is hyper U. Hyper U. What is hyper U?
Hypo is low, hyper is high.
Yeah, good. You got it. So hypo, let me be in the camera. So hypo is minus, low. Things go down. Hypo, things go down. Hypoxemia, oxygen went down. Hypoproteinemia, protein content went down. Hypoglycemia, glucose went down. Hyponatremia, sodium went down. Hypo is things going down. The opposite is when things go up, and that is excellent. That is hypertension. Hypertension. Hyper. So hyper is when things go up. Hyper when things go up. Plus, plus, plus. Hypo is a problem. Hyper is a problem. It's not the more the better. Hey, hyper, the more the better. The more glucose, the better. The more oxygen, the better. The more carbon dioxide, the no.
Has to be done.
There has to be excellent a balance. What do we call the balance? Homeostasis or U.
This student, Elena, was looking for you, but I think she mispronounced the name, so I said I don't know the person.
Yeah, I think so.
It was me.
That was you?
Okay.
My apologies.
No
Happy New Year, everyone.
It's my mentor, Dr. Paul.
All right, no.
Is this hot in here?
I think I have a remote.
Okay, so we're doing just fine. Hyper, we don't want that. Hypo, we don't want that. We want U. So that is homeostasis. Very good. That is homeostasis. So your body is always living in a state of fighting hyper, fighting hypo, and remaining at U. Remaining at, we don't want hypercalcemia, not hypocalcemia, eucalcemia. Not hyperglycemia, not hypoglycemia, euglycemia. Not hyperoxemia, not hypoxemia, euoxemia. Not hypertension, not hypotension, eutension. So there are built-in mechanisms in your body that restore your homeostasis. I remember I asked one of the students, what is homeostasis? And the student said, homeostasis is balance. Homeostasis is balance, and homeostasis is keeping a steady state. Well, that could not be any more wrong. Because if I'm in the North Pole to be balanced, that means the North Pole is 0 degrees. I'm 0 degrees too. I'm dead now. So that homeostasis does not mean balance. Homeostasis means you keep a constant internal state no matter what happens on the outside. So the outside pushing you to be hyper, you stay at U. Outside temperature is so hot, 30 degrees Celsius. You're sweating, sweating, sweating. You're at 25 degrees Celsius. Outside is so cold, 15 degrees Celsius. But you are shivering, 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 25 degrees Celsius. So here we saw two opposite things. Somebody is so hot, sweating, 25. Both of them. You put a thermometer in their mouth. Both of them 25 degrees. But one is sweating and the other one is shivering. One is sweating, the other one is shivering. So that shows you the sweating is the way to achieve homeostasis. Yeah, for that individual. It's cold, you shiver. You shiver in order to achieve homeostasis. So it is your bodily effort to resist the change, resist the outside hyper. It's always a fight between outside, inside. People around, peer pressure. That's a psychological stress. This is an environmental stress. Hyper is a stress. That's a hyper stress. Cold is a stress. Hyper stress and hypo stress. Even in psychology, around you, bad influence, peers, peer pressure, pushing you to do hyperactivity that is harmful. So you resist that. Then the opposite. You're living in a society where there is no pressure for hyper, there is pressure for hypo. You just be lazy, you do nothing. You don't get up, you don't do no work, you just chill. But you resist that. So your capacity to resist is always a fight between outside and inside. They are never at peace. When they are at peace, you see the dead body, the cadaver, you touch it, it's just like the outside. They are at peace now. The cadaver is now at peace with the external environment. The external environment won. But the external environment did not win because your body is constantly fighting energy expenditure for you to achieve the glorious state of being in homeostasis. Oh, it is costly. Being in homeostasis is not cheap. It requires energy. It requires that you do metabolism. It requires a lot for you to remain here in this seat, you. That's why the dead body could not achieve you no more, so it went into shock. So you must understand that your body will undergo stress, either hyper stress or hypo stress. They are as bad. No one is better than the other. Your body is constantly being poked around. Stress. So your body is going to fight that stress with adaptive, with built-in adaptive mechanisms to help you overcome the stress. And most of the time, you win. Most of the time, you return to homeostasis. But trust me, sometimes, if the onset of the stress was so fast, you went outside, you opened the door, it's freezing cold, you dropped it. So fast, the speed was so fast. Number two, the duration. I mean, it's you outside, it wasn't that cold in the beginning, but you stayed outside for 4 hours, 5 hours. Now the duration is a lot. So if the stress onset was quick, if the duration of the stress was long, the time of the stress was long. Or number three, the intensity. The intensity was so severe. I mean, outside hot in the summer, 40 degrees. But I mean, imagine you outside is 500 degrees. Now that's really intense. So the intensity of the stress, intensity, the power of the stress. Sometimes if the stress onset was so fast, the duration is so long, the intensity is so powerful, these guys fail. They say, I'm sorry, we got overwhelmed. Oh body, we cannot help you. We cannot achieve homeostasis no more. We got overwhelmed. The intensity was so severe. The duration was so long. The onset was so quick. We are overwhelmed. It's too much now. Then your body will collapse. Your cells will undergo necrosis. Necrosis. Now this is language now. Necrosis. Necrosis is when the cells die in a very vicious, debilitating, injurious way. Necrosis. Oh, you don't want necrosis. And then there's apoptosis. Apoptosis, oh, we love that one. Apoptosis. Apoptosis is good. We love death by apoptosis. Apoptosis is when the cell that is damaged is gonna, it's called programmed cell death or programmed cell suicide. So the cell will kill itself in a programmed manner. It will chop up into blebs, B-L-E-B-S, into blebs. So it will become like bubbles, balloons. The big cell is going to rupture into little circles, little balloons, little harmless blebs. You're going to be little harmless blebs. But in necrosis, no blebs. In necrosis, the cell membrane is going to rupture. The cell membrane is going to rupture. And all the intracellular content is going to leak. You're going to have leakage. The lysosomes is going to spill out. It's like a nuclear power plant. You know, in the nuclear power plant, everything in a container. But when a spill, you have a nuclear spill, what's going to happen to the neighborhood around that place? That is what's going to happen in necrosis. So that's what happens when we get sick. When somebody goes coughing, it is because the lung cells are necrosis. They are collapsing and the plasma membrane rupture, lysosomal enzymes spilling out and damaging nearby tissue. But in apoptosis, that will not happen because they're going to be like balloons, blebs, harmless balloons. We want that. But apoptosis is costly. Apoptosis wants oxygen. Apoptosis wants metabolism. Or it's going to cost you to die healthy. But necrosis is so cheap. So when there is gunshot wound, that's necrosis. When there's bacterial infection, that's necrosis. Blood coming out, sputum, that is necrosis. That's not apoptosis. So always was associated with the pathological damage. That is going to be necrosis. So you don't want that. But you must understand disease begins with stress, hyper or hypo. The next step is cell injury because the cell was overwhelmed. So number one, we have stress, followed by, after stress, what do we have after stress? After cellular stress, we have cell injury. So now the cell is injured because the pain and the intensity and the duration and the onset is so severe. Then we move on from cell injury to cell death. If the injury was manageable, we die by apoptosis, and that is a good positive death. Otherwise, if things are really bad, we're going to die by necrosis. And necrosis is minus. That's negative. That's harmful. That's really bad. Then after necrosis, Then after necrosis, we're going to shift to inflammation. We're going to shift to inflammation. And inflammation is gradually, you begin with acute inflammation, the first stage, then finally chronic inflammation. So the stage of this, it didn't begin overnight. It began a long story: stress, cell injury, cell death, necrosis, acute inflammation, chronic inflammation. So it's a long, gradual steps, negative, bad steps that led me to where I am. Yes.
Question, between necrosis and apoptosis, which one of them, what's the duration time that I'm cell death? Because I know with some necrosis, it basically takes a long time for like, the flesh to rot and stuff like that. So I just wanted to know what's the duration.
Okay, so the duration for apoptosis is longer because the cell now has time to clean up, turn into harmless blebs. It's very costly. You know, it's like a U-Haul truck come and you pack things up and you package things. So the cell is going to be packaged into harmless blebs. So that one is going to take a long time, and the cell is going to dispose of itself properly, get rid of itself properly in a way that doesn't harm the surrounding. But in necrosis, no time for that. That's why we have necrosis. So necrosis happens because the onset was so quick, duration was so long, intensity was so powerful, the cell ruptured. Everything vomited out. The nuclear power plant spilled, the nuclear spill. All of the dangerous, toxic, is now spilling all over the neighborhood. That's exactly what happens in necrosis. All the harmful substance is leaking to the nearby.
That's the skin also.
For everything. So when we say cell, all the cells. Heart cell, blood pressure, necrosis. Stroke, that's necrosis of the brain. Okay, trauma, when you have trauma. Even infection, bacterial infection, that's also gonna cause necrosis. Every time, but you have a lot of tissue, that's why it doesn't get as bad. But when you get older, the more you get sick, the more necrosis. The more you get sick, so that's why you got to be careful of the flu. You got to be careful of the asthma, because when you're young, it really doesn't mean anything. But when you get older, more lung tissue dies. More muscle tissue dies. So this is what we're going to be doing in anatomy and physiology. We're going to be looking at what makes each system different, what makes each cell different, what makes the job done, the function, the physiology, the anatomy, the structure, what enables, what is the relationship between the function and the structure, what is the relationship between the cell and its function, and how are all these different bodily systems do their job. Okay, and then we're going to be looking at this very important concept of regulating the stress, and how does all your bodily systems work so efficiently to restore homeostasis, to ensure that you have no, to ensure that you do not have any form of debilitating necrosis or inflammation. So the first lecture is we're going to be defining what is anatomy. So anatomy is the study of the structure. Okay. I stepped one out 1 minute. Everybody is hanging out. Yes.
First, I was curious about listening.
Yeah, everything is recorded. Don't worry. Yeah,
You're gonna upload it?
I'm gonna upload it. Yeah.
See?
I'm not gonna throw all of this at you. Don't worry. Okay, guys. So step number one. Are you guys following me on the lecture slides? So you can go to the modules and click on week one. Week one is gonna have that document. So you can follow on the screen also. So anatomy, what is anatomy? Anatomy is the study of the structure. Why do the cells look the way they do? By the way, all the cells, even though they look different, but they do have the same structures. They all have three compartments. This is really, really important. When you look at the cell, you have to understand that when we begin anatomy and physiology, we're not immediately going to begin with the system. We're not going to begin with the endocrine system. We're not going to begin with the blood system. We're not going to begin with the cardiovascular system. We're not going to begin with the respiratory system, the immune system, and the lymphatic system. We're not going to begin with all of these different systems. Why? Because we're going to begin with the cell. So first we begin with the building block. We begin with the cell and we work our way upward. So we begin with the cell. That's the basic thing. The basic thing is the cell. That's the basic unit of health, is the cell. The basic unit of disease is the cell. The basic unit of life is the cell. The basic unit of death is the cell. We are alive because the cells are alive. We are dead because the cells are dead. We are healthy because our cells are healthy. We are sick because our cells are sick. So multiple cells give us tissues. And multiple tissues give us organs. And multiple organs give us organ systems, network. And organ systems give us organisms. And multiple organisms give us population. Population. That is reproductive biology. So now we're going to look at the cell, the structure and the function of the cell, the anatomy and physiology of the cell. Then we're going to move up the next floor. Structure of the tissue and the function of the tissue, then structure of the organ and the function of the organ, then structure of the organ system and the function of the organ system, then structure of the organisms and the function of the organisms, and then the structure of the population and the function of the population. So we're going to go in order because they build up on each other. Another thing very important for you to know, this chronology in the book is not, they do not put the endocrine system after the nervous system randomly by rock, paper, scissors, because that's the actual order. You can never understand the endocrine system unless you understood first the nervous system. You can never understand the nervous system unless you first understood the musculoskeletal system. You will never understand the musculoskeletal system unless you before that did the skin and the integumentary system. Then you're going to before that learn, so you're never going to understand the tissue unless you got done with the cell. You're never going to understand the organ unless you finish the tissues, then the organ, then the organ system, then the organism, then the population. You cannot shuffle through. It's not going to make sense. So when we begin with the cell is the smallest unit. But that does not mean this cell has nothing inside. So when we look at the cell has three different compartments. Compartment number 1, and compartment number 2, and compartment number 3. What is compartment number 1 for the cell? That's the cell membrane. That's the cell membrane. That's compartment number 1. That's the cell membrane. This cell membrane is a universe. It has phospholipids. It has protein ion channels. It has enzymes. It has cholesterol. It has carbohydrates. Don't think it is just the fence of the cell. There's a whole galaxy happening in there. There is laboratories studying parts of this plasma membrane. It is a galaxy. The next part is known as the cytoplasm. The cytoplasm is divided into two parts. The first part is known as the cytosol, and the other part is the organelles. The cytosol is a colloid. It has water, jelly, has a water, gel, and solutes, and electrolytes, and salts. And this is the liquid part of the cell, the swimming pool that the cell, in the swimming pool, they have the in the swimming pool, they have the plastic toy that they throw in there. The rubber duck, yeah. I'm looking at. I thought, what kind of animal is that? Dragon or what is it? Duck, yeah. It's a duck, the rubber ducky. So all the organelles are the rubber duckies, and the cytosol is the swimming pool water. So all the organelles are bathed in this cytosol. So now we don't have one rubber ducky. We have multiple rubber duckies. We have the mitochondria, the powerhouse of the cell. We have the Golgi body. And we have the lysosome. We have the peroxisome. We have many organelles. We're going to get through all of them. But they are all the rubber duckies in the cytosol. The last part, what is it? Nucleus. Excellent. Very good. Very good job. Excellent. The nucleus. And your name is? Samantha. Excellent job, Samantha. The nucleus. These are the three universes of the cell. This is where all the activity is happening. And obviously inside the nucleus, who do we have? Our DNA. That's where the genetic expression happens. The genetic expression. That is where the genetic expression happens. So this is now we're going to learn about the structure of the cell and the function of the cell. So anatomy is the study of the structure of the cell, and physiology is the study of the function. So anatomy and physiology are complementary, and they are never entirely separable. They are dependent, they are interdependent. And physiology provides the meaning to the anatomy. So anatomy is what makes physiology possible. Okay, so what are the ways to examine the structure of the human body? Back in the day, they thought that anatomy was just what you see. They thought, what exists is what we see with our eyes. We see a cow, there's a cow. We don't see bacteria, there's no bacteria. Then they realized when they discovered the microscope that there is microscopic anatomy and macroscopic anatomy. And they realized that actually microscopic anatomy is even more than macroscopic. If you look at planet Earth from outside with a satellite, you will never think there's human beings in there. If you didn't already know that we exist, if they just show you a picture of planet Earth, will it ever occur to you that there's people walking around? No, it just looks like you can see big Africa, big North America, big South America, big Asia, big land, but you never know there's these little things. Same thing. If you look at your arm from outside, you never know there's bacteria and parasites living in there. They are just like us. They're living in there. Maybe they're going to school and doing all that kind of stuff in there. They're going to nursery school. They're doing they're living. That's their community. That's their life. but you would have never known because you can't see it. The same way you are microscopic when you're looked at from far away. They are the bacteria and the parasite and the worms and all the microorganisms. They may get offended if you tell them you're micro. So, oh, you can't see us, but we're not micro. The same thing if somebody tells you from another planet, we never thought you're so small. I'm not so small. You're just far away from me. You can't see me. So this now revolutionized the discovery, the greatest discovery that ever happened. The most valuable invention that ever happened is the microscope. Because the microscope took us to universe galaxies that we never thought they existed. That our vision is nothing. Like what we see is actually 0.00009% of what really exists. You cannot see every little thing that's out there. And thank God we can. I mean, imagine you can see all of these kind of things. You get heart attack. Okay, so now we're gonna look at these two things. The anatomy, the structure and the function of the cell and everything in the cell. We're gonna be studying the macromolecules, the micromolecules. We're gonna be studying the tissues. Then we're gonna move upward to study the big things, the liver. But it's gonna be a long time before we do that because we gotta be and you gotta focus really well on the little stuff because the little stuff is what will make the big stuff make sense. Understanding the cell. People are so eager to learn about the musculoskeletal system, so eager to learn about the endocrine system, and nobody want to care about the cell anatomy and physiology. It will never make sense unless you focus really well on the cell. And you go in order because they build up like a pyramid. They build up. If you slack and you go past one topic, you don't spend a lot of time on it, everything is like a dominoes effect. Everything else is not going to make sense. So you really have to have deep attention. So the anatomy for the stuff that we can see, the macroscopic anatomy, we first do inspection. Inspection is when, to make observation. It's very important to make, your mom sees you. Oh my God, you are so red today. What happened? Are you sick or something? Oh my God, you're so hot. Do you have fever? So the senses is what guide us to make an observation. Because if you cannot sense, you see somebody screaming in the street for help, you heard that. If you couldn't hear, you will never hear the screams of somebody need the fire truck, and you can call 911 for the fire truck. If you cannot feel or see the fire burning, you cannot make an observation. So observations are always based on senses. That's why the dead body can never observe anything. It is when you're alive, you can sense, you can observe. It's too hot in here. Like, doctor, how come I didn't notice it was too hot? I was too busy talking. So you observe with your senses. That's why you got to work on that's why the best, most brilliant, intelligent are the people who have intact senses. Okay, this drinking the alcohol, all this is gonna destroy the senses badly and make an alcoholic liver disease and make your vision, make your hearing, all this smoking the weed and all of the heroin and all of this dangerous stuff is gonna make you insensible. It's gonna make your heart not soft and you're not gonna be detecting things. So in order to be brilliant, have good analytical, critical skills, you have to ensure that your senses are intact and they are because without them, you cannot observe. You cannot be a doctor and you just not paying attention. The patient comes in, many times to the clinic, a patient comes to me complaining of one thing. I notice they're not even complaining about it. I notice it. I inspect it. I say, listen, what you came for, that's 10%. There's another 90% we need to deal with. And then, we take care of that. But another doctor, okay, what do you need? You swipe the insurance? Okay, goodbye. Okay, so you have to have, as nurses, your senses have to be really intact. And then after inspection is palpation. And when you use the palm, you use the palm to feel if there is some sort of swelling or to feel if there's temperature. or if the patient is gonna scream if you just touch them so lightly. Auscultation, you see, that's another sense. You hear, there's a murmur, a heart murmur. The heart's supposed to say lub dub, lub dub. Okay, your heart, does it say lub dub? It does. Then you're fine. Let me hear his heart. The heart says lub dub drup, lub dub drup. Uh-oh. That's heart murmur. So your senses, you're always training your senses to be on pinpoint so you are able to make an observation and detect clinical signs and symptoms that even the patient himself or herself not even aware of. You get to detect it for them on their behalf. Percussion is when you tap on the body and you feel for the resistance and listen to the emitted sound. So you tap on the body. And this is, by the way, this is obsolete. This is back in the day. Now we do ultrasound. Now to see if there's something, if the liver is enlarged, hepatomegaly or splenomegaly, nobody's going to do that. Unless if you are in my country, then they don't have over there the technology, especially in the villages. So you still use those. So it's really not obsolete because one day I believe we're going to go back to these kind of days. You know, this industrial society that we live in, the gas and the petroleum and the airplane is all running on fossil fuel. And it's going to run out. Trust me, 100 years, 200 years, it's going to run out. How are you going to drive an airplane with no gas? It's going to be finished because they're using it like crazy. They're using it for the airplanes. They're using it for the cars, for the trains. Maybe not in our lifetime, but one day it's going to be gone. So all of that stuff is going to be like the dinosaurs. The people are going to come after and say, oh, they used to take this to fly all over the world. And yeah, it's going to be gone. So now we're going to go back to percussion again. So you must know these and sometimes you are in a situation where you don't have the technology. You're in an airplane, you're far in an island, so you need to master the skills so to save somebody's life. Dissection is when we cut the cadaver, and we have different ways of cutting. I mean, imagine you have a boiled egg, and you just look at it from outside. You don't cut it in the middle. Will you ever find out there is yellow yolk in there? Unless you cut it, slice it open, you will never know. So there is different, we're going to learn different techniques in lab of how to cut. Sagittal cut, coronal cut, transverse cut. Each one has a significant way of finding something out. There's no one cut. You have different types of cuts. Then there's the comparative anatomy, and that is the study of multiple species to learn. So we compare different animals to humans. And that's very significant because now they're doing this xenograft. You know, xenograft? You know, back in the day, they had the autograft. They take one part of your body, of your skin, and plant it on another part where you had a burn. That's autograft, from yourself to yourself. And then they had allograft from another fellow in your own species. It's got to be a human being like you. That's known as allograft. And lastly, there's the xenograft, because the baboon heart, sometimes the pig heart, they plant that. So now, if it wasn't for comparative anatomy, we would have never had xenograft because we just keep ourselves only in our human species. But sometimes in life, when you go in different directions, in different trajectories, good things will come to you. You're going to open yourself new opportunities. So now when we investigated animals, we were able to relate something helpful to the human beings. There's exploratory surgery, and that is opening the living body to see what is wrong. And radiology is the branch of medicine specializing in the imaging. Gross anatomy is when we look at major things that we can, macroscopic, we can see, that's gross. Histology is microscopic anatomy. Microscopic anatomy is the examination of tissues with microscope. You cannot see. And that part is a lot bigger than what you can see. If you say there's a lot of animals, wait till you look in the microscope. There's a universe in there. Histopathology is looking at for the mechanisms that lead to the creation of disease processes. Cytology. Ology is science, or the study of, the learning of. Sight is the cell, is the science of the cell. That's what we're going to be doing for the next couple of weeks. Cell, all about the cell. So the study of the structure and the function of the cells in fine detail, known as ultrastructure. So physiology is the study of function. Sometimes we specialize physiology. We do neurophysiology, endocrinology, histopathology, hepatophysiology. So you choose a system and you just stick to it forever. And you just go learning deeper and deeper and deeper. Applied science. You apply all the science to that particular certain organ system. The same way we had comparative anatomy, we also have comparative physiology. So comparative physiology is the study of different species to learn about their bodily functions. So we look at the hearts of other animals and we look at the functions of these other animals. So it always very important to look at the history of anything you're getting into. Anatomy and physiology. Some people think it's boring. Some professors actually skip this first lecture. You know, all of this history. Oh, it's very important. You gotta learn that they used to do experiment on slaves. Okay, you're a slave, you have no right. Let's do experiment on you. They used to do slavery on they used to do the people that not slave but they have death penalty. They are free people. They have death penalty. They go and they say, well, might as well use you for some science experiment. So now we're gonna learn in bioethics that we cannot do that. And you're gonna see that people used to study. They suffered. I mean, to get to the point where we are now with the anatomy and the physiology, you're gonna see that the information they learned, like back in the day, they used to do cadaver, but they had no idea about the formaldehyde. So they couldn't remove all of they didn't know about embalming. So they couldn't remove. So the cadaver is putrefied, sitting in there, putrefied and smelling bad. I mean, some students used to just faint just by the stress of that nasty, putrefying smell. But some of them, they just kept through because they want to learn. And then they came up with really valuable information. And, they tried, they had some theories that we now laugh at. Like, some people say that, the heart pumped the blood to the liver and the liver pumped the blood to the brain and stuff like that. And then the brain pumps the blood to the rest of the body. Some of them had things like, this part of your body is where God put this and that and this kind of stuff that is not scientific. But there has been an evolution. And the first woman, it's very important to understand the first woman in science, that was Metrodora. Metrodora was a Greek woman physician. She published a textbook on the obstetrics and gynecology. So she was really deep into the birth and labor and saving mothers who are giving birth to children. So that's like the mother of medicine, Metrodora. And then there's Claudius Galen, and he would be the physician for the gladiators. I mean, back in the day, they didn't have the basketball in the NFL, so their fun was to watch two people killing each other. Yeah. So after the fight, when actually that's actually happened now, UFC, all that kind of stuff. Yeah. So that's still. But what Claudius did was he would wait till the fight is over, then he would take samples of the injured body and investigate all of the dead tissues. And he came with ideas that about what could be a DNA, that what is now known as DNA. He kind of sensed it back in the day. Then there's Maimonides. This is the Jewish father of medicine. He did some very influential contributions to medicine in times where the church banned science and scientific investigation in Europe. Maimonides was able to find contributions. Then there is the Muslim father of the medicine, that is Avicenna, and he wrote the canon of medicine. So you see that there is a holistic worldly effort to push this anatomy and physiology, and to give us some ideas about what is anatomy and physiology. Okay, I'm not gonna ask you about any of that on the exam. I'm not gonna ask you maybe I'll ask you who's the mother because that's one is important, of the medicine. You say there's Metrodora. What specialty was she into? Endocrinology, neurology, obstetrics and gynecology. So you choose that one. Okay. Now this science that we're going to investigate, this science that we are investigating, we first have to define what science is. What is science? Science is the study and the knowledge of the laws and the truth that govern the particular specialty of interest that we're looking at. And when you say science, there's different kinds of sciences. There's life science, there's the science of the living things, the biology, there's the science of the non-living things, there's the physical sciences, then there's the science of the society, the social science, the behavioral science. So what is that? What is the social? So we have science, we have science, we have science, and science is divided into, first of all, life science, life sciences, then there's physical sciences, and lastly, you have social sciences or behavioral sciences. Life sciences are the sciences of the living things, and that is biology and the microorganisms, what we see with the microscope, and then there's the macroorganisms. Then there's the physical sciences. These are the non-living things. The stars, the planets, the astronomy, the archaeology, the fossil fuel, the geography. Is that living?
No.
That is all non-living things. Then there is the social science or behavioral sciences, where we study the incentives, the rewards, or the penalty, and the motivation, or the punishment that influence human behavior. So, for example, why are you here today? Because there is a reward and there is a penalty. A reward is you're gonna come here today, you're gonna have credit for showing up, for attendance, you're gonna get credit for participation, you're gonna get credit for doing the labs, and you're gonna get credit to your grade and you're getting closer to A now. There is another, that's a short-term goal. The long-term goal, you're going to be fulfilling your nursing degree, and then you're going to be getting paychecks, and then you're going to be fulfilling your life calling of becoming a nurse and helping those who are suffering in disease. So that's a reward. Penalty. What's the penalty? You don't show up here, come here today. 0, no points. You're gonna get closer to an F, and now you are, not gonna become a nurse. No paychecks and no fulfillment of your life ambition and helping and making a difference in your society and helping those who are sick. So you see, your behavior is guided by two things: reward, incentive, plus good things, and minus things, punishment. Okay, and the criminal says, why should I go rob a bank? I'm going to make $1 million, but if they catch me, I'm going to be in jail for 30 years. So they think otherwise. So always the penalty and the reward. That is what guide human behavior. So people who misbehave is because they believe the punishment is not high and the reward is too high. But if sometimes we change and reinforce the behavior with making the reward high and the punishment high, so that nobody gets to commit those wrong misbehavior. So that's social science. So these are the sciences. So now we looked at the different types of sciences. Now, when we look at science, what is science? What are you looking at? This one?
Yeah, I want to see what's about.
Archaeology, fossil fuel, and geography. That's just an example. Science is the knowledge and the study of the knowledge and the study of the truth and law of truth and the law of the systems we're looking at. So, for example, life science is looking at the truth and the knowledge of the truth and the laws of the living systems. Physical sciences is looking at the truth and the law of the non-living things, the astronomy, the stars, the planets, the archaeology. And the social sciences, these are the behavioral sciences where we study the incentives, the rewards, the penalty, and the punishments that influence the behavior, not just of humans. Sometimes we study bird behavior. Sometimes we study animals' behavior, mating behaviors. So what leads animals to act in these kinds of ways? So study children behavior in psychology. But in the end, it's a science. That means we have to study the laws and the truth of it. And how do we do that? Is by the scientific method. We achieve the studying of the scientific method. So the scientific method is a procedure, is a uniform procedure, is a uniform. This is known as the scientific method. The scientific method is a procedure for us to investigate the laws and the truth that we want to know about. So we must first, in the scientific method, have an observation that is based on what? What did we say? The observations are always dependent on what? Excellent. Very good. So your senses. So for example, you start your car in the morning. The car is not starting. You observe that. So when you observe something, you immediately have what? You have an observation. So then you're going to have a hypothesis. Hypothesis. Then you never have a hypothesis. The hypothesis, what does the hypothesis say? My car is not working because the battery is dead. Or maybe my car is not working because the transmission, the engine is not working. Or my car is not working because there's no gas. The gas tank is empty. How many hypotheses are those? Well, those three hypotheses. So now do we stop at a hypothesis? You just okay. Well, the hypothesis is the battery is not working, or the engine is failed, or there's no gas. And you just stop there. No, you're gonna take the next step, and that is the investigation. Investigation. This hypothesis that you made was testable. This was a testable. This was a testable hypothesis. You can test if the battery is working or not. You can test that. You can test if the gas tank is full or not. You can test if the transmission is working or not. So the hypothesis has to be what? Testable. Number two, the hypothesis has to be falsifiable, meaning it can be proven wrong. So that means if your first hypothesis was that the battery is not working, you take your battery, put it in somebody else's car, hook it there, start the car. If their car started, that means your battery is good.
Plan B.
So that was plan B. So that was testable. You tested it, and it was falsifiable because your hypothesis, the battery was not working. You put in somebody else, it worked, so it was falsified. It can be proven wrong. You were able to prove it's wrong. So plan A failed. Hypothesis B is not working because the gas tank is empty. You go check out the gas is full. Now what's left? Something wrong with the transmission. So now you look at testable and falsifiable hypothesis. Now imagine somebody starts the car is not starting, and they say, oh, I know why the car is not starting, because God is not happy with me. Maybe it's true. It is maybe true. But is it scientific? It's not. Somebody say, my car is not working because, oh, they did black magic on it. Maybe it's true. They put the ghost on it. Maybe it's true. We're not talking about it's true or not. We're talking about is scientific or not. Can I test if God is mad at you or not? That's why the car is not working. Can I test if they did black magic on it? I cannot do that. Maybe it's true, but I cannot test it. Now we're just focusing on science. But it doesn't mean somebody say there's a haunted house. Like, okay, maybe it is haunted, but I cannot test that scientifically. Okay, so the hypothesis has to be testable. You cannot say black magic or God or all of this because you cannot test that. It has to be falsifiable. You cannot choose something that you can't even prove it's wrong. I mean, if the car has no battery, you cannot say because the battery is not working. I cannot prove that wrong because there is no battery. And then you move on to the investigation. So you investigate with no bias. You do not give a medication that you want to test that is going to bring down the heart pressure. You bring all patients who have high blood pressure. You say, listen, this is going to drop your blood pressure. Just take it easy, okay? And just sit over there and don't make anything. And I don't want your blood pressure to go up. Now you're influencing the results. Just give them the blood pressure medication and tell them be the way that you usually are. Leave them like that. And then you're going to get the right result. No bias. No bias. No bias. Then after the investigation, you're going to get your results. So you put them nicely in a table. Then after that, you're going to discuss the result. You interpret them. Oh, yeah, you're going to compare them. And then you're going to have peer review. You're going to have experts. Your other class, your other professor, other scientists, they're going to read your experiment and say, oh, you missed that. Because we're human beings, we have memory that is faulty, irreliable. We make mistakes, so you need somebody else to point you in, oh, you missed that one. You will definitely, no matter how finely you try to craft something, you will always need counsel. It's always best to have like experts to make decisions based on counsel of the expert, not the decision based on what they say on Facebook or on the YouTube or you go to the proper place. What is inductive versus deductive? Inductive versus deductive. What is inductive? Inductive versus deductive. Inductive means we work our way up from the bottom to the top. That's inductive. We go from the bottom to the top. Deductive is upside down. We deduct. We go from top to the bottom. So what does that really mean? In inductive reason, we have observation. We have observation, and from the observation, we make a theory, or from the observation, we make a theory or we make a hypothesis. For example, back in the day, they didn't really know how the brain works. So they knew that the brain eats a lot of sugar. The brain utilizes a lot of sugar. That's why people who study a lot are usually obese, because they're always snacking on sugars. Because when you're studying, your brain is going to be utilizing a lot of sugar. So now what they did was they mixed sugar with radioactive, shining chemical substance that when you look at it in the computer, it's going to shine. It's not going to hurt you. It's just going to shine in the computer. And they brought a volunteer. They said, listen, we're going to inject you with sugar that is radioactive. And guess what they did? They put a helmet that is attached to EKG, and that shows the brain. That shows which parts of the brain light up. That means that part is utilizing glucose. And they tell them, please do us a favor. Look at this following picture. So they look, there is sandwiches and the burgers and the pasta and the lasagna, and then one part of the brain lit up. That part is utilizing a lot of glucose, is burning a lot of glucose. So they say, oh, that part is responsible for appetite. Otherwise, it wouldn't light up, because he was looking at food. Then they said, okay, so it's something else. And they show him all the sexual, all the seductive things, the sexually infatuating, the sexually enraging things. And he's looking, then another part light up. Now they say, oh, that part is responsible for sexual reproduction. Then they say, please do us a favor, start walking on a treadmill. Then he starts walking, then this part starts to light up. So, oh, this is the region that is important for coordinating the movement and locomotion. Then they said, we'll give you a math problem, solve the math problem. So he solved the math problem, then the other region lights up. So that is an example of inductive, because they observe which region light up. Then they decide that is the region for this. You get it? So inductive reasoning is when you rely on you observe that's the region that lit up. So then you make a hypothesis, that is the region responsible for this. That's the region responsible for that. Clear?
Clear.
Now, what is deductive? Deductive is you don't work with no observation. With deductive, you have no observation. With deductive, you dealing with facts or premises. You're dealing with a premise or a fact. Meaning what? All mammals, why do we call them mammals? Because they breastfeed the baby. Breastfeed milk to their baby. That's premise number one. Number two, human beings are mammals. Then you deduct that human beings breastfeed baby. Did you observe anything here? There was no observation here. You deducted. You deducted based on premises. The premise that mammals breastfeed babies. Then you were told, by the way, human beings are mammals. Then you do 1 plus 1 is 2. Human beings are mammals. Mammals breastfeed babies, or human beings breastfeed babies. Whales are mammals. Whales are mammals. Mammals breastfeed their babies. Then whales are breastfeeding their babies. So that is deductive. Deductive is when you don't rely on observation. It is when you rely on a premise. You rely on an established principle, a fact, and you base upon it. This is so important. You knowing inductive, because I'm going to ask you that on the exam. I'm going to ask you which of the following correctly depict the difference between inductive and deductive reasoning. And the answer is inductive reasoning relies on observation to help us make a hypothesis. But in deductive reasoning, we make a hypothesis not based on an observation, but rather based on an established premise or a fact. Scientific, yeah. Is one better than the other? Is one superior? No, it's different. It's different way. Okay. Falsifiability, we talked about that. Then the experimental design, there's got to be a sample size, and there's got to be a number of subjects in the study. There's got to be a control group. That group resembles the treatment group, but does not receive the treatment. So in that blood pressure medication, you have the same group. All of them, they have blood pressure, but you give them pill full of water. You give them M&Ms. You don't even know about it. And then you see. So you cannot just, it can be falsifiable. Otherwise, if you don't have a control group, you don't have falsifiable. Psychosomatic effects. We'll take a break in 10 minutes. Psychosomatic effects. These are effects of the subject. So next week, you guys said you want to start at 1:30. We'll leave earlier, like an hour earlier instead of 2:30.
When did that 1:30 become effective?
I'm sorry?
When did that 1:30, when did that?
Okay, so you weren't here.
No, I wasn't.
Okay, so we said the following. According to the syllabus, we start the class at 2:30. If you would like to come at 1:30, we can begin then and leave an hour earlier. If you can only. If you cannot, then we must start at the time that the class begins. Yes, you had to stand up.
How does that affect, lab?
Yeah. I teach both. So we're gonna do both.
You come at 1:30, I can't do that.
Yeah, so we're supposed to be here 2:30 to 8. If you come 1:30, we'll leave 7.
I can't do that.
Okay. So yeah, if you cannot do that, then if you cannot do that, then it has to be we all have to agree as a class. We cannot begin before. We cannot begin before some people. Raise your hand if you cannot. Okay, because we have to accommodate everybody. We have to really accommodate everyone. So raise your hand if you cannot come here at 1:30. Okay, so 2. Okay, so how about this? Let me consult. I'm going to consult the administration. I'm going to consult the administration and see that maybe what can we do? Because if the overwhelming majority can start 1:30, we will see if we can come up with something fair for everybody. And if you come late, then maybe the lab you can get to do it online or something. But we will see. We will see. Okay. All right, guys. So we will have our break in 10 minutes. The psychosomatic effects. The psychosomatic effects. Yes. Yeah. Lab is no. If we shift upward, you're gonna leave earlier. But if we start 2:30, it's gonna be 8. I'm sorry. If we start an hour earlier, we will end an hour earlier.
So 6:30 because That's the chronic, with like what period.
Yeah, okay, yeah, if we start an hour, okay, okay. If we start an hour earlier, we will shift upward an hour earlier. That's all. Okay. Okay, guys. Yeah. If we start an hour early. Okay. Psychosomatic. Okay, guys. We need to continue. The psychosomatic effects. These are the effects of the subject's state of mind. So, and that is tested by giving a placebo. So, what's the significance of giving placebo, M&M or water? But placebo is you tell them, you lie to them. You tell them, listen, this water pill is actually the blood pressure medication. Actually works on some people. Some people even start to develop side effects. So it happens. It happens. And people thought that the doctors thought that they are maybe imagining that they have side effects, but actually they did. Some of them vomited, some of them had diarrhea, some of them they had increased breathing rate, some of them they had they had
Were
different.
they allergic to it?
It was water.
Oh,
It's just a psychological thing. Yeah, it's funny. Okay, statistical testing. So these are statistical tests to provide statements of probability that the treatment was effective. We talked about the peer review. So natural selection is how fit are you to the challenges. So the theory of natural selection um, explains how the species originate and how they change through the time. And they help us to understand the human body. 1.4. If you look at that, yeah, 1.4. So this influential biologist, Charles Darwin. So if I ask you on the exam, maybe about somebody, it's got to be him because he's the father of this um, yeah, the theory of the natural selection. It explains how the species originate and change through time. Sorry? The what?
The for the minds, these theories.
Yeah.
Are we going into them?
No, we're just like
Okay.
going over, but we're not gonna go in deep.
Okay.
When it comes to the exam, I'll give you guys a study guide. So this study guide is going to, if you focus on the study guide, I'm going to be giving you a study guide. I'm going to be giving you summary videos that you're going to watch the video, summarize it, upload it for credit. And also the study guide, you're going to solve it and upload it also for credit. No plagiarism is going to be searched by Turnitin. So to make sure that you can work together, you can discuss together, but the final copy has to be unique like your fingerprint. It cannot be matched. The answer may be the same, but has to be worded differently. The answer is the same, but you must come up with a way to add some information that makes it in the engine not detectable as the plagiarism.
You're doing our DNA now.
Yeah. Okay. Alrighty. So this shows you now walking upright, known as the bipedalism, and that is standing. So these are characteristics we developed in the natural selection that helped us achieve, that helped us to achieve the ability to survive in the challenging environments that we are living at. Um, and we talked about this, that the molecules, we have the cells, but below the cells, there is a whole universe. There is the molecules. Remember, we talked about the salts and the ions and the electrolytes and the enzyme proteins that we get from the euchromatin. These are the ones who make the cell living. So don't think just because you have a house, does that mean you have no heater, you have no AC, you have no fridge, you have no carpet, you have no sofa? So you have all these things that make the house what it is. Obviously, the smallest unit in the society, in the neighborhood, is the apartment or the house. But the house cannot just be on itself. It must have micromolecules that makes it what it is. So we said that the cell have organelles. The cell has a cell membrane. The cell has a nucleus, and the cell has all the rubber duckies and all the pool and all the cytoplasm and all the molecules. So all of those are very important for our. So this is the hierarchy of complexity. This figure is going to be on the exam. You're going to see that my questions on the lab, I'm going to give you the actual question for the lab exam. I'm going to give it to you. So you're going to go into the book. Like, for example, for this one, if you look at this, figure, actually, let me show it to you. Let me show it to you. Just give me a second, please. Just I'm going to show you exactly how. There we go. Okay, just give me a second. Okay, there we go. Okay, this is an example of. Okay, can you please look? That's an example of the lab one exam study guide. Thank you. Thank you. So look at this question here and look at this figure. So in this figure, that is figure 1.5 in the textbook. So what you're going to do is you're going to go to the study guide and you're going to see. You see the study guide? It has the letter I, it has the letter H, it has the letter G, it has the letter F, E, D, C. So you see A, B, C, D, E, F, G, H, I. Then you go into the book and you look into the figure, this one. This doesn't have A, B, C. It has actually what they are. So your job is to look for each letter, this letter, to identify all the letters. Not only identify them, you read in the textbook anything that has to do with this figure. You will see my question on the lab exam. That's not the lecture, the lab exam. Exactly that. You will see that right there. And you will see the same figure, but you don't know what the question is. That's your job now. I gave you the figure. I gave you the textbook. So you're going to go into the textbook and find that figure and look at all the letters. I understand the letters. Remember, this is structure and function, anatomy and physiology. What's the structure? What's the function? What this figure is trying to tell you? What are these things? You're going to come on the exam. You're going to find the same figure, and now you're going to see the question. Okay, so that's how it's going to be for the lab. For the lecture, it's going to be like that. It's going to be like this. Anatomy and Physiology 1 lecture exam 1 study guide. It is due at 11:59 p.m. You're going to see a list of all the questions. You solve all these questions without putting the question. You just put the answer so that in turn it in, it does not, you have like same question with everybody. You solve it, you solve all the questions, and then you submit them, you study them. I'm going to look through them, I'm going to grade them, and then when you come on the exam, you will see that the questions, they were very similar to the study guide, to the homework, to what I'm saying in lecture, and you're going to see that the lab is those figures, which are also be similar to the study guide, also similar to the homeworks. Yes.
So the study guide is a homework assignment.
Hmm?
The study guide is a homework assignment as well.
Yeah, the study guide, you can look at it as a homework assignment, but yeah, because you have to post it. You have to submit it to the portal that I'm going to open for you. And I'm going to give you also the multiple choice, right or true, the multiple choice questions. You have to choose the right answer and you focus well on them because they also will be very close to your exam. So don't just, the students, they do them, they Google them and take photos and sending in group chat and all that kind of stuff. If you do not study these questions and you don't understand them, you're going to see them on the exam or a question really similar around it, reworded, and you're going to get it wrong. So you understand the question, why is each answer the right answer? Why is each answer the wrong answer? And you will see that my study guides are, and my homework questions, and what I do in lecture, they are like mirror images. So it pays a lot to show up and pay attention and do the study guide, do the homework questions, and you will see that you will do excellent.
Out of the book, right?
Yeah.
Questions for each chapter.
Don't, if you want to do those, it's up to you. But yeah, it does have it. But if you just stick to what I give you, that's all you. So begin with that. You want more information, do that at your own leisure. But ensure that what I give you comes first. Then expand on that later. So if you do that, you will do excellent. Most of the students in my previous course, they got A's because they just did the assignment that I told them on the time. You did the study guide, you do the homework questions, you study the study guide, you study the homework questions, you do the summary, the videos. Told you I'm going to give you video summaries, you're going to watch them and summarize them. This is anatomy and physiology. This is not just any other course. This is going to build, make you or break you. And you do those assignments, then it's gonna really help you a lot and do well. Okay, so I'm not like trying to make the exam challenging, or it's just very straightforward. You solve the questions, you will do well on the exam. Okay? Yeah, it's on the syllabus. Okay. All right, guys. So let us now take a break and we'll meet in an hour to do the lab.